Hi folks, my name is Cole, and I have a Master's of Immunology. Today on Investigate, Explore, Discover, we're going to be looking at a new oral antiviral drug, Molnupiravir, and its phase 1 clinical trial. So hang around with me through this whole video to get the down low on all that you need to know, so that way we can dive into some exciting experimental results. Plus, if you look in the description below, there are instructions on how to get a free NFT. Now, there's one major reason why this particular topic is timely. That is due to the ongoing COVID-19 pandemic. COVID-19 is the disease caused by the virus SARS-CoV-2. This respiratory illness spread incredibly rapidly around the globe. To give you an idea of how rapidly, this disease was first reported in China at the end of 2019. By the beginning of 2020, it was reported in 4 Asian countries, and in the short course of a month and a half, it then spread to 28 countries worldwide. It was declared a pandemic by the World Health Organization on March 11, 2020, and 2 weeks after that, it was found in almost every country worldwide. Since then, there have been over 265 million cases of COVID, and it has been a contributor in over 5.2 million deaths. Now, to compare that to other forms of mortality in the United States, in 2020, COVID-19 was the 3rd leading cause of death behind cancer and heart disease. To put this in the perspective of major historical events in the US, by now, COVID-19 has contributed to more deaths than both of the world wars combined. And this graph is a little old, but COVID has also now caused more fatalities than the 1918 Spanish flu. So what exactly does COVID look like in those who contract it? Well, there are many symptoms, the most common of which is body aches, fatigue, loss of taste or smell, coughing, or fever. But in some instances, this infection is not so mild. Serious infections result in trouble breathing, persistent chest pain or pressure, or even confusion. Since this disease is so new, the long-term effects of infection are still currently unknown, but more information is being revealed every day. There are ways that you can mitigate your exposure to the virus and reduce the chance of serious illness occurring. You can wear a mask in crowded locations or stay home if you're sick. It also helps to wash your hands frequently, not touch your face, and stay 6 feet away from people where possible. Now, getting vaccinated also drastically lowers the likelihood that you will experience severe complications with COVID. SARS-CoV-2 itself is a coronavirus with a single-stranded RNA genome. It is made up of many proteins that allow it to survive in its environment to enter into and infect host cells. Now, like all RNA viruses, it undergoes a replication cycle that goes through multiple stages. Each of these steps is a whole process in and of itself, and disrupting any of them leads to an antiviral effect. Today, we are going to focus on the inhibition of RNA viral genome replication. This can be done by blocking the activity of viral polymerase or incorporating a competitive substrate that wouldn't normally be there. This induces errors in the replicated RNA. These errors can cause lethal mutagenesis for the virus and prevents it from completing its replication cycle. And this is what Molnupiravir does. Well, not Molnupiravir itself, because it is technically not the thing that is mediating this effect. Molnupiravir is a prodrug, which is quickly cleaved in plasma to EIDD 1931. After distribution to various tissues, EIDD 1931 is converted to 5' triphosphate by host kinases. This 5' triphosphate is the competitive alternative substrate for the virally encoded RNA-dependent RNase polymerase. And upon 5' triphosphate incorporation into the newly growing chain of viral RNA, it induces errors resulting in viral error catastrophe. Viral error catastrophe is the idea that by increasing the virus's error rate beyond a biologically tolerable threshold, this causes impairment of viral fitness, leading to viral extinction. Now, previous studies of Molnupiravir found that it was highly effective at treating seasonal and pandemic influenza infections in a ferret model. Molnupiravir was also able to effectively prevent transmission and viral replication of SARS-CoV-2 in ferret, hamster, and mouse models. Now, due to the preliminary success of Molnupiravir in animal models, it was given the thumbs up to be tested in humans. Testing new drugs in humans requires rigorous testing procedures, which involves multiple phases of verification. Clinical trials are composed of 4 different phases to ensure the safety of patients. Phase 1 of clinical trials, which we're focusing on today, utilize a small number of healthy participants to assess the safety and tolerability of a drug. This encompasses looking at a drug's pharmacokinetics and, if possible, its pharmacodynamics. Now, pharmacodynamics studies how a drug affects an organism. So this looks at the activity of the drug in the body. Pharmacokinetics studies how an organism affects a drug. This involves looking at a drug's absorption, distribution, metabolism, and excretion. Now, some parameters that are looked at are the highest concentration of the drug in the blood, the half-life of the drug, and the lowest concentration in the blood. The area under the curve that these parameters create signifies the overall drug exposure, which can be compared across different dosing levels. Phase 2 of clinical trials assesses the therapeutic effect of the drug, the optimal dose, and the toxicity profile. This step is the first time that the drug is tested in people with the disease that it is intended to treat. These trials are blinded and are done in hundreds of people. When the data is collected from this step and the treatment shows efficacy and minimal negative effects, this allows for testing in phase 3. This phase of clinical trials confirms the safety and efficacy of the treatment in a larger population, usually in the range of thousands of people. Now, if the drug is confirmed to be safe and efficacious against disease, then it will be approved by the FDA. This allows the drug to be given to the public. Once given to the public, there is still ongoing monitoring for safety surveillance and further optimizations. Now, I want to take a moment and really highlight why antiviral discovery research is so important. Right now, we are experiencing the COVID-19 pandemic, which continues to influence almost every aspect of our daily lives. Creating a safe and effective treatment for those with severe cases of COVID-19 could help to reduce the burden of disease worldwide. However, to produce an effective treatment, it first needs to be proven that it is safe for human consumption. If you think that these are some important reasons to research this topic, go ahead and tap the like button. This brings us to the paper that we're focusing on today. This paper is called Human Safety, Tolerability, and Pharmacokinetics of Molnupiravir, a novel broad-spectrum oral antiviral agent with activity against SARS-CoV-2, by Painter et al. From Ridgeline Therapeutics. In this paper, the authors evaluated single and multiple doses of molnupiravir for safety, the effect of food, and the drug's pharmacokinetics. This study was a phase 1 randomized double-blinded placebo-controlled study in healthy volunteers. The authors of this paper first got together a group of over 100 people for this study, the demographics of which were predominantly white men around 40 years old with a healthy BMI. The test participants were then randomized in a 3-to-1 ratio of people taking molnupiravir versus the placebo in the single and multi-ascending dose part of the study. For the single dose trials, the subjects were given one of an ascending dose of molnupiravir. For the multi-dose trials, subjects were given one of an ascending doses of molnupiravir twice daily for 5.5 days. Now, subjects from both of these trials were then monitored for 14 days to assess safety, tolerability, and pharmacokinetics of the medication. The authors also tested in a 1-to-1 ratio the effect of consuming food before taking 200 milligrams of molnupiravir or not eating beforehand. Throughout the study, this drug is being administered in an oral capsule, with the exception of highly concentrated doses over 800 milligrams, which a liquid state was used because it allowed greater flexibility in single-dose administration. Speaking of single-dose administration, overall, 24 of the 64 subjects, 37%, receiving the single ascending dose reported an adverse event. There was no apparent dose-related trends, and the single group with the most adverse events reported were those who were given the placebo. The most common complaint was a mild headache. There was no serious adverse events and one moderate headache amongst the placebo and molnupiravir groups. Now, in the cohort of people that were given molnupiravir twice daily, 25 out of 56, or 44.6% of people, reported adverse events. There was one report of moderate oropharyngeal pain, pain in the extremity, and influenza-like illness. Otherwise, all adverse events were mild, with the most common report being of diarrhea. One person discontinued the drug use because of the emergence of a treatment-related rash, which was resolved following discontinuation of the drug and a topical steroid treatment. Now, in the food evaluation trial, there were only 3 people that reported mild adverse events and no serious events. The authors also found no clinically significant findings or other dose-related trends in clinical laboratory vital signs and ECG data, and there was no changes in hematological parameters. Furthermore, dose escalations were discontinued before a maximum tolerated dose was reached, because plasma exposures reached higher than efficacious levels based on scaling from animal in an influenza model. Molnupiravir actually gets metabolized pretty efficiently. Most of the samples collected from people had concentrations of molnupiravir below the limit of quantification and really only showed up after large doses were administered. That being said, the active drug EIDD 1931 was abundant in plasma samples. When participants were given a single dose of molnupiravir, the maximum concentration of EIDD-1931 typically occurred 1 hour after consumption in doses up to 800 milligrams, and the half-life of the drug in plasma was around 55 to 75 minutes, which declined in a basic monophasic pattern. However, at higher concentrations of 1200 and 1600 milligrams, the time to reach the maximum concentration was delayed. The dosing of EIDD-1931 and the area under the curve, however, followed a dose-proportional pattern. This dose-dependent pattern was also observed during clearance, with smaller doses clearing slower. However, this did not change that the majority of the drug was excreted after 4 hours. Now, when people were given multiple daily doses, it appeared rapidly in the plasma with a median max concentration achieved between 1 to 1.75 hours at the beginning and end of the treatment. These plasma levels declined in a monophasic pattern with a half-life of, again, 55 to 75 minutes, and the dosing of EIDD-1931 and the area under the curve again followed a dose-proportional pattern. This dose-proportional pattern persisted during clearance and did not change that in all samples, most of the drug was cleared after 4 hours. Now, what was particularly interesting is that after the 6th day of administration, it consistently revealed a biphasic clearance pattern at the higher doses of 600 and 800 milligrams. This consequently increased the EIDD-1931's half-life to be seen in some patients over the course of 24 hours. What is also important to note is that even at the 6th day mark, there was no evidence of drug accumulation. Now, a pretty common occurrence when taking medication is that people eat beforehand. So the authors looked at the effect that eating has on taking this medication. The authors found that eating delayed the onset of the maximum EIDD-1931 concentration and decreased the maximum value. However, the area under the curve balances out with the higher, faster peak from the fasted state. Now, both of these states allowed for clearance in a monophasic fashion, and EIDD-1931 remained detectable in the plasma between 9 to 15 hours in the fed state and 6 to 9 hours in the fasted state. To quickly summarize everything altogether, the authors of this paper looked at the safety of molnupiravir in humans when administered orally. They tested increasing doses and found that it had few adverse events, was well tolerated, and had no dose-dependent toxicities, and was converted into its active form, EIDD-1931, quickly throughout the body in the study's healthy cohort of participants. Now, not only do I think that these safety findings are exciting to investigate and learn about, they are also significant in a broader context. This information is significant because it shows that molnupiravir is, number one, rapidly broken down into its active form, EIDD-1931, as well as being well tolerated, with fewer than half of the subjects reporting adverse events. This means that testing can continue on to phase 2 of clinical trials. Now, all science is basically a stepping stone for new knowledge, and these steps are driven by questions. And I had a few questions myself when going through this information. My first question is, will the safety profile seen in this phase 1 clinical trial be replicated across different ages, sexes, ethnicities, and BMIs? Furthermore, probably one of the most immediate questions around a new antiviral is, how effective will it be in humans, in particular at fighting COVID-19, the annual flu, or even MERS? Now, if this treatment turns out to be effective, how well could it then be incorporated into our toolkit to be able to end this blasted pandemic once and for all, so that way we can get back to our lives that do not revolve around the hysteria associated with new COVID infections? And as always, my final question revolves around you. What sort of ideas or questions popped into your head when hearing about this information? I would love to hear about it in the comment section below. Also, let me know if there are any topics that you'd like to hear about in the future. Ultimately, I hope that you learned something, but more importantly, I hope that you enjoyed your time doing so. So if you did, give this video a like and subscribe for more in the future. Well, that's everything for today. Thank you for watching, and I will see you next time.